Good morning, Anuradha madam. Good morning, Aparna madam.
Yes. Shall we wait for a few minutes because we said we'll start at 11 o'clock. I think still 5 minutes time is there. You can tell that the chat option will be there in the last and the feedback form will be posted at that time and all. Yeah, yeah. Then we sure. So total 540 registrations are there all over the country, sir. I think till now 314 people have joined.
Excellent number. Almost it amounts to 65–70%.
Yes, sir.
Very good number.
Because this is aimed both for students as well as faculty, ma'am, sir.
Okay. What is the proportion of students, Anuradha madam, in the law?
I have not yet checked, but it is 50-50, sir. Because I think, Aparna, do we have any numbers like that, faculty and students?
We have faculty 70% and students 30%.
Okay. But PG students, they are participating, and of course, even undergraduate students are there. Will we start? Is it time?
Yeah, I was hoping that there will be more students because I thought that in the sense that it's okay, absolutely, but I'm just saying that because I thought that there are a lot of things that you can tell about, especially the master students, where they can, if people who are interested in coming for research, many a times they come and ask that what should be their career option.
Yeah, ma'am, definitely students, we know that they enter right in time. That is at 11 o'clock, 11:05.
Okay. And that's absolutely
So
okay.
their entry will be after 11.
I'm sure that 30% is going to spread the word. And
Yes.
yeah, because the kind of situation we are in, we really need a workforce who will be, involved into a lot of research. So main idea was to excite them about it.
Yes, ma'am.
So you have to instruct me how I once again share my.
Yeah, yes, ma'am. After you share, Aparna?
Yeah, ma'am.
Ma'am has to annotate her slides.
Yes.
Before after you share in ma'am, go for annotation. So you know that the toolbar, the taskbar is there on the top, and then enable participants.
Okay.
So that they will not scribble anything on your screen.
1 second, ma'am. Sharmistha, ma'am.
Yeah.
Once our Anuradha ma'am and Ashok sir speaks, I will do stop sharing. Then immediately you share the screen.
Okay.
When you move your cursor at the top, there is more option.
Yes.
Okay. In that more option, if you click, there will be an option called disable annotations from the participants.
Okay. Those 3 dots.
Yeah, 3 dots. Correct. 3 dots under that more will be there. In that,
All
you
right.
do the disable participants. That remaining all will be fine.
Sure.
Thank you.
Let's start the webinar. Very good morning to one and all. I am Dr. K. Anuradha, Head Department of Microbiology, Bhavan's Vivekananda College. Extend a very warm welcome to all the participants for today's webinar on antibiotics, why crisis. Before I start, I wish everyone are healthy and comfortable stay at home in this COVID pandemic time. I'm sure we are effectively utilizing this time. And I'm glad that the Department of Microbiology and Bhavan's College announced series of webinars. We thank our management, and we also thank scientists and professors and academicians for immediate response, and they are rendering their services to the student community through this webinar, which is very appreciating aspect, ma'am, because all the professors, the moment we ask, they are coming forward to help the student community. I'm sure through this webinar, students are going to be benefited. And today's webinar is on antibiotics, why crisis? I'm sure we all know the antibiotics usages, and several million people are using the antibiotics because they are saving our lives. And I don't think any individual is existing without usage of antibiotics. And they have both medicinal and non-medicinal uses. And we know that the repeated usage of these antibiotics led to the concept of drug resistance. And even the non-medicinal uses had shown large impact on human health. So this webinar is going to address at least some of the challenges that are associated with the antibiotics. I welcome Dr. Sharmistha Banerjee, resource person of this webinar. Good morning, ma'am.
Thank you. Good morning to all.
Yeah, before I introduce the speaker, may I request our principal, Professor Y. Ashok, to address the gathering.
Good morning, everyone. And good morning, Dr. Sharmistha, madam, for being with us.
It's a pleasure.
Radha, Dr. Aparna, and all the delegates. I wish the webinar a grand success. And the Department of Microbiology has always been on the forefront in doing such activities. They are the first research center in the college. They are one of the leading departments which are doing a lot of research, writing good articles. Led by Dr. Anuradha, Department of Microbiology is doing a lot of good work, and they have a passion for research. They have a passion to promote science, and thanks to Department of Biotechnology and initiatives taken by all the departments led by Dr. Anuradha, we are granted the status of Star College by Department of Biotechnology, MHRD, and all such initiatives will definitely be very useful to the students and also the faculty. I'm happy a lot of students are participating in the webinar, and especially as far as antibiotics go. Even I have a dilemma. Sometimes say antibiotics is the cure for everything, and sometimes they say excessive use of antibiotics is a bane. So as a common man, definitely we have our own dilemmas and doubts. I am sure Dr. Sharmistha will throw a lot of points to ponder on the issues relating to antibiotics. I wish Dr. Anuradha and her team all the very best for the success of the webinar. Good luck, Anuradha. And thank you, Sharmistha.
Thank you so much.
Thank you, sir. May I request Aparna to focus the slides so that change the slide, please. Aparna, you muted yourself. Professor Sharmistha Banerjee she's from Department of Biochemistry, University of Hyderabad. Ma'am's area of interest is molecular pathogenesis and immunology of Mycobacterium tuberculosis, human immunodeficiencies of Mycobacterium tuberculosis, and HIV co-infection. Dr. Banerjee received her PhD in biochemistry in 2005 from University of Hyderabad, and her postdoctoral studies at the Laboratory of Transcriptional Biology on mechanism of row-dependent termination of E. coli. She worked at Center for DNA Fingerprinting and Diagnostics, Hyderabad, and she also worked at Molecular Cardiology Unit, Lerner Research Institute, the Cleveland Clinic, USA, on role of integrins in prostate cancer metastasis. Presently, she is professor in the Department of Biochemistry, School of Life Sciences, SU. She teaches immunology and enzymology along with various topics on infectious diseases and host pathogen interactions. there are many credits to honors and credits. She received Chancellor's Award in 2017 by University of Hyderabad, and she's a recipient of National Women Bioscientist Award in Young category in 2016 from Department of Biotechnology, Government of India. And she's a recipient of Professor B.K. Bachpat International Travel Grant for Young Scientist in year 2018. And she also bagged Innovative Young Biotechnologist Award in 2011 from Department of Biotechnology. And she received BioAsia Innovation Award in 2008. In 2005, she received INSA Young Scientist Award from Indian National Science Academy. Her research profile is very, very appealing and attractive. And she has published more than 41 publications to her credit. And she published in 41 papers in national and international journals. She completed 4 research projects under various schemes of UGC and DBT. She is a recipient of various grants from different funding agencies like INSA, DBT, DST of worth 55 crores. Total number of citations of her publications is 737. I think I show very soon she is going to reach the more than 1,000. And her H-index value indicates 14. Again, here we have to keep in mind her citations and her H-index clearly indicates how much quality research she is putting in for the scientific community. So with this brief introduction, I welcome Professor Sharmistha Banerjee to interact with the, to start with the proceedings.
Sharmistha ma'am, I'm
over to Dr. Sharmistha Banerjee.
Screen.
Thank you so much, Dr. Anuradha. I think I'm ready to share my screen, and thank you so much for calling me for this webinar. I'm looking forward to interact with all of you.
Yes, ma'am. Ma'am, immediately after you share screen, annotate your screen, ma'am, so that nobody will scribble on your screen.
Sure.
On more.
So I can still see the other screen.
Ma'am, I stopped share screening.
You can start, ma'am.
You can start share screening.
So I still see your screen, or no? Okay, how do I?
No, just move your cursor down and you
Share.
will get share screen
Share.
option.
At the lower panel, right?
Yes, ma'am.
Okay, lower part.
Lower panel.
Participants, you please keep your video off so that you can completely concentrate on the lecture.
Ma'am, now go to more option, ma'am.
I have disabled. Can you just check?
Ma'am, your screen is
It's
visible
okay.
to all of us.
Yes, ma'am. If you have disabled, that's enough, ma'am.
But can you see? Can you see my screen?
Yes, yes, we can see,
Okay.
see your screen as well as you.
All right. So I think I can start now.
Yes,
So
ma'am.
thank you so much for calling me to this webinar. I'm very excited about it, about interacting with the topic, which is being fairly discussed, and in a situation of what pandemic we are having, possibly it keeps coming back about antibiotics. Although we talk about antivirals more now, but antibiotics have been there with us for many years now, and eventually we have started seeing something what we call as antibiotic crisis. And I thought that I'll share some of my views here, some of our concerns that we all should be aware of, and at the same time what all areas of research going on in this is especially for students who are often think about picking up an option of a research career, and they want to know what all areas are exciting at the moment. And so I'm going to put some of my views regarding that. So antibiotic, when we say why the crisis, possibly, I am not able to change my slides.
Ma'am, use your computer arrow marks, laptop arrow marks.
I'm doing that. Okay. So the crisis, when I say why the crisis, possibly are these kind of news where we hear about superbugs. Very recently, they're all over the newspapers, and you people must have heard about something what is called as New Delhi metallo-beta-lactamase 1, which was found in one of the microorganisms, which source was supposedly India and Delhi. And the reason it was called a superbug because that particular plasmid gave a resistance to a lot number of antibiotics, and it could not be treated by conventional methods. Not only that, we keep on reading about news where we say that fungal infections which were otherwise fairly could be at least treatable are now becoming untreatable by days. So including warnings, do not know how true they are, that sometimes they can even resist the alcohol sanitizer. And not to say about the drug resistance TB that we keep hearing about, where India have a huge load of the drug resistant TB. So the way I'm going to have organized my presentation is that we'll quickly and very briefly go through about the discovery of antibiotic to just appreciate the work that has gone to reach up to the stage where we are, and of course understand how they work very briefly, so that we also know that if they do not work, what are the things that we should think about. We also require to understand that why there is a sudden surge in emergence of antibiotic resistance, and more important, how have we arrived there? Because if we know how we have arrived there, possibly there is a way of controlling it, regulating it, and not to forget that emergence of antibiotic resistance possibly is going to stay with us. So it is best that we handle it so that we know how to best manage it. Then we'll come to some of the mechanisms by which bacteria develops resistance, and after we have gone through this whole very brief outline of it, I would like to spend some time, and because I've noted down certain points, that to tell students mainly that how we are fighting it and what are the areas that are the active research area where people are trying to contribute in various forms to tackle this problem of antibiotic resistance. So when we talk about antibiotics, the first thing that comes to our mind is about infectious diseases or communicable diseases. And if you go through, I try to get as recent statistics as possible, but if you go through any statistics in any top 10 causes of death, possibly especially in low and middle income countries like ours, you will find a range of antibiotic communicable diseases or infectious diseases that are the major reason for the cause of death. They cause morbidity, mortality, and definitely have a huge economic burden in our country. To give just an example of tuberculosis, where we see that there is a huge burden in 2018 itself. We registered almost 26 lakhs of tuberculosis, and not to say 4.4 lakhs deaths. And amongst the cases, there were 1.3 lakhs of multiple drug resistant TB. The reason I'm thought of just saying, giving you statistics like this is we are talking about a disease which is curable, a disease for which we have antibiotics, and yet we have statistics, yet we have numbers which definitely are not acceptable. So just to tell about antibiotics, as we generally know about antibiotics, is if we segregate the two, the thing antis against bios means life, it would mean that anything that can stop the growth or kill microorganisms. So that would mean bacteria, fungi, viruses, protozoa, etc., etc. So, however, in medical dictionary, generally, or WHO, the way antibiotics are defined, generally are told as it is against bacterial infection. So if I'm talking about antibiotics, I'm primarily trying to tell about antibacterials and not antivirals or antifungals, which are definitely there. They're also in a larger definition antibiotics, but antibiotic by definition means antibacterials. So in short, the reason I'm trying to tell you this is that when you get a virus, a flu or something, and somebody is trying to give you antibiotic, most of the time antibiotics do not work against viruses. You require antiviral to work against viruses. So possibly it is important for us to distinguish that. So antibiotics have both naturally occurring molecules as well as a lot of molecules that are synthetic. That means we have a lot of antibacterials which have natural sources as well as it has a synthetic source. So if I give you an example of rifamycin, which is from an actinomycetes source, and the analogs of it is used for tuberculosis treatment. And if you also recall, especially these for the students, you have chloroquine, which is another derivative of quinine, which is against from a bark of cinchona tree. So these are natural sources, and so these molecules which have an antibacterial activity occur in nature. And also there are synthetic molecules which have been synthesized by man, like sulfonamides, like quinolones, that are actually synthesized, but they also have antibacterial property. They target bacteria and they can kill or limit their growth. So when we say that antibacterials have a natural source, most of the time the discovery of antibiotics have decided on experiments like this. So in several, even now, a large number of antibiotics are being attempted to be identified by isolating the sources or the natural sources, and a very doable way of doing it is to grow two cultures together and see if they can grow together or if they have a relationship, what we call as antagonism. So if you see these culture plates, you have here Aspergillus niger and Bacillus subtilis being grown, and there is a clear zone where you have a clear zone where there's no growth possible, and you can also see that there is a precipitant here that would suggest the first step towards identifying possibly a molecule which has the potential to restrict the growth of yet another organism. And this practice is fairly recent, and we still do, and you will find many papers where people are trying newer and newer species by co-culturing them. For example, the second figure shows that there is a specific isolate of Streptomyces, which is being trying to be worked out if it works against different pathogens of fish. So in aquaculture, you also require certain very specific antibacterials so that the aquaculture is economically can be grown. So in short, these molecules which are being released out is a natural phenomena, and a phenomena which is microbial antagonism, which is the basis of the modern use of antibiotics. And it was recognized by no one other than Louis Pasteur, who recognized it long time back that there is this natural phenomena that is given by nature to us, which has a lot of secreted molecules and possibly for the ecological existence or coexistence of several organisms. Just to very quickly do, we understand how penicillin was discovered and by Fleming, and followed by that was Paul Ehrlich, who made an organo arsenical compound which was used against syphilis those times, and syphilis was very, very severe. It was not treatable. It used to cause deformities, and it was a huge threat. And this was a breakthrough in the concept of chemotherapy, which was followed by a synthetic drug, Prontosil, which was the forerunners of all the sulfur drugs that we have today. And Domac was, of course, he earned a Nobel Prize, but was asked not to take it by Germany at that time. But I think he is very well deserved, because from here began the concept of chemotherapy against bacteria. Since then, from this very, very scary era of which preceded antibiotics, there was started a very golden age of discovery of antibiotics. And I do not know whether I have populated this slide possibly sufficiently or not, but you can see that there are several years I've discovered. With every year, the new class of antibiotic has discovered, and which of course is even continuing now, but we do see that there is a dampening, or there is a slowdown in the discovery rate. And so all these different kind of antibiotics eventually were found to be of majorly, they had two kind of properties on the mode of their action. As you know, the one which does not permit the proliferation of bacteria were called bacteriostatic, and amongst them are very familiar antibiotics like chlorphenicol, erythromycins, sulfonamides. Where are there other antibiotics which can kill the bacteria and what called bactericidals, and that includes a lot of beta-lactams and other molecules. And also, in those discoveries they found that there is a wide spectrum as well as there's a narrow spectrum of antibiotics. It's very simple, fairly simple. The definitions are fairly simple and doesn't make us very difficult in understanding that a wide spectrum would mean that it would be able to attack a large number or large variety of bacteria. Is it good or bad is something which we require to think about, but definitely when it comes to controlling very resilient, very difficult bacteria, the wide spectrum antibiotics are a good idea. But there are also narrow spectrum antibiotics like isoniazid and polymyxins, and there are intermediate spectrum which also has a few lesser range than the broad spectrum antibiotics. So antibiotics here. So to just tell about what we know about the antibiotics. So how did these antibiotics work? So as if I have to think about that, I'm going to find a molecule which will not let the other organism grow. It is but naturally you require to target something which is very crucial for the bacteria. So many of the time, the antibiotic discovery were done, or the metabolites or the discovery of molecules are tested by something what we call as a disdiffusion assay, or sometime you might have heard about the dilution. You do grow the bacteria in presence of different concentration of that particular extract or that particular metabolite or that particular molecule, synthetic molecule, and try to see, and eventually try to see the growth kinetics, and eventually try to figure out that which of the physiological process that it is trying to attack. And if you take a look of the list of antibiotics that are available, because I'm not going to go really through the whole thing, you will find that they have been targeting very major physiological processes. The cell wall of bacteria defines its entity, and many of the antibiotics, they target the cell wall synthesis. Amongst them are beta-lactams, vancomycin, bacitracin, and also polymyxins, which target the cell membrane here and not the cell wall here. So entity, when it is lost, it tends to it can lose its osmolarity regulations and can burst and it can die. Similarly, many antibiotics, they target very specific metabolic processes. Some of them are here listed here, like folate synthesis. We have sulfonamide, which is important for synthesis of including nucleic acid synthesis. You have specific enzymes that are targeted, like DNA gyrases, which can unwind DNA. You also have RNA polymerases, which are targeted by many of the molecules here. Rifampicin is given as an example. Protein biosynthesis has been continuously a target, and there are not only the one which are listed, there are a number of them which actually inhibit protein synthesis very specifically for the bacteria in by several molecules, either by attacking its 50S or the 30S subunit of the ribosomes. So all these and possibly more, method, not only that, I think possibly these days people are trying to find out the metabolic network and trying to identify those critical enzymes which are what we call it as choke points of bacteria, which I'll be talking about a little bit later when we talk about new antibiotics that are being attempted to be discovered for replace or tackle the emergence of drug resistance. So antibiotics are broad spectrum, antibiotics are narrow spectrum, they can be bacteriostatic, they can be bactericidal, and they definitely attack a very important physiological event of bacteria and not of the host, and therefore are the one which takes care of the bacterial infection. So all this was going very well, and suddenly with the introduction of antibiotic, there is a death due to infectious diseases started coming down tremendously, and I could not get the data beyond that, but you can already see little bit of surge from when it is trying to reach 2000, and not only that, there was also an increase in livestock. You'll be surprised to see that huge amount of antibiotics, in fact, over 10 times more antibiotics are used by either in livestock or in agriculture than to treat people. Okay, so all of this is because of not only taking care of their infections, but sometimes they also have a mass boosting or effect, which there's a rampant use of antibiotics in livestock started. So all was going good, and there were huge economic benefits that started coming. And this is where the problem begins. Till it starts that we started getting these news. There is an extreme drug-resistant tuberculosis. We call them, the tuberculosis has first-line drug, second-line drug, and then third-line drug. So first-line drug, you become resistant, the bacteria becomes resistant, treated with second-line drug, which has huge toxicity. Then the second-line drug, it becomes resistant to, then it has to be treated with very limited number of options, and it becomes an extreme drug-resistant tuberculosis, and almost with very little treatment available for such kind of conditions. We have such Staphylococcus aureus infections which were resistant to methicillin. They were found in, then there were NDM superbugs reported, and all kind of resistance or emergence of antimicrobial resistance bacteria, it started. So sudden surge of multi-drug resistant strains. Now we have two timelines. I was just talking about a very happy timeline where there were a lot of antibiotics that were periodically discovered. And then we also have a timeline that periodic discovery of antibiotic, there is also a periodic resistance to such molecules. And if you see, there is a huge increase in the frequency of the development of resistance to this molecule. And that is where our concern is. So the idea is that how did we get here? We have discovered a molecule to take care of our health, and eventually we have lost it. And that possibly is because of a lot of antibiotic abuse. So first, let me talk to you about certain personal things or very individualistic things that we quite often do without knowing that we are contributing to emergence of drug resistance. So many a times, and quite especially very much in India, we take them when they are really not needed. Such examples can be even if you have a common cold or something and you're just feeling malaise, it is very, very common to have a quick course of antibiotic, which actually doesn't do anything other than you getting possibly loose stools or diarrheal-like conditions, because it does have an effect on your gut. They are continued sometimes when they are no longer necessary, and the reverse also happens. Suppose there's a course of 5 days given, and after 3 or 2 days you start feeling better, and then eventually we ignore the rest of the doses thinking that it is not required really. So sometimes given in wrong dose, sometimes the infection, which can be actually controlled by a very narrow spectrum agent, generally a broad spectrum agent is given. And possibly it is given because many a times people do not really want to know the kind of organism that has caused the infection. They are not really, they are not really diagnosed for whether it's a gram positive, it's a gram negative or something. And so if you give a broad spectrum, it just takes care of everything. Most important and most frequent is possibly the self-medication, which and not completing the process. So that this keeps happening at a very personal level, and possibly one thing that we should be really be worried about and also be careful about is not to tamper with what has been prescribed by the doctors. So doctors do have a huge responsibility here, but that is one of the major reasons why we see that there is a huge increase in the frequency of emergence of antibiotic resistance. Well, these kind of images are quite common, and you may have read more reports than me that effluents from various kinds of companies, which also includes antibiotics, are thrown into sources. And from there, they start seeping into groundwater. From there, they start seeping into their biota. And eventually, you see that the antibiotics are somewhere where they are not supposed to be. Not to mention about the abuse that is done in poultry and livestock. And this is something which we actually don't even think much about. And only of late, I think we have started awakening to such kind of problems. Just to give you an idea, there is some of the reports says that to produce a kg of beef, around 40 mg of antimicrobials are used. Around 145 mg of antimicrobials are used to produce a kg of chicken. There are surveys in 2010-11 that was done in many of the aquaculture. Of course, the antibiotics were used both for prophylactic as well as curative purposes. And they have found they're rich in things like oxytetracycline, ampicillin, formalin, and various kind of ceprofloxacin, things which are in the aquaculture. And when they have taken out the specimens from there, they found they are very, very rich in these antibiotics. So so what does it mean to us? All good. So we have, we started treating our infections by discovery of antibiotics. And eventually we thought that antibiotics can take care of many things. And there was a rampant uncontrolled use, fair enough that we do see all these things. So what does it mean to us? Does this molecule accumulate somewhere? Do they get magnified? Just like we are opening to something called global warming, and that has been quite a few years now, I think we should open up to the fact that these antibiotics are just not pollutants. They do get accumulated, they do reach us. So a process called biomagnification, which you all know, that it is a concentration of any toxin, and it can be a pesticide, it can be an antibiotic in tissues of tolerant organism at successive higher levels, trophic levels in a food chain. That is how we define biomagnification. So are these, whatever I'm talking about, do you think that if we think that these are hypothetical situations or these are not real, then we should wake up, because there are very concrete scientific evidence towards the existence of such problems. For example, so I would say antibiotic bioaccumulation problem and the consequent increase in emergence of pathogenic bacteria in nature are real problems. And I've just exemplified a few papers here which are very recent, where they have found that in this particular study, they have tried to find out the various kind of, there are 9 sulfonamides, 5 fluoroquinolones, and 4 macrolide antibiotics that they looked for. And they found that there is an increase, especially in case of sulfonamides and fluoroquinolones, which are very frequently used for our treatment, are accumulated in almost all the 10 fish species or even some of the invertebrates in this particular study. So all these, and there are many, definitely show that they do accumulate. Now the question again comes, of course, if you go through these, they definitely say that it has not come to an extent where it will cause a harm to, because of consumption, there can be a harm in the, to human. Of course, it doesn't say, they say it has not come to that level. But that would also mean it's a kind of warning that if we do not really are careful about it, they're like global warming. This can also become a huge problem for us, and it is a good idea to be, to wake up to such problems before they actually become unmanageable. So all this accumulation, so what does it mean, and what does it actually lead to a junction of resistance? When we say antibiotic resistance of a bacteria, we mean to say that a bacteria which was earlier getting killed by a particular antibiotic is no more getting killed or restricted by that antibiotic. Now, these kind of accumulation, for example, in this particular study, it is, I think it's it's a very recent study in which people have tried to find out that beyond the disease control of animals, sometimes animals are often given antibiotic for growth promotion and all. So these people, they studied the presence of antimicrobial resistance genes in a milk metagenome, and they investigated their genetic position and their linkage to mobile genetic elements. And what they find is that the samples that they studied, it contained cephalosporins, cefamycins, fluoroquinolones, peptide antibiotic, tetracycline, and even they had certain PC1 beta lactamase, which actually can be a mobile element, and that facilitates the transfer of resistance genes to other bacteria. And that other bacteria can be in our gut as well. So what I'm trying to say is that the one which gets into the environment, the one which gets into, finds its way to our body in some form of generating antimicrobial resistance. Similarly, you will find also many other reports where they have said that when the antibiotics are dumped into the environment, they are along with other pollutants, there is also an accumulation of features in these natural biota about resistance to multiple antibiotics, because there can be a process of co-selection. So, in short, what we are trying to say is that when antibiotics are not used optimally, when they are not used in a place where they are required to be used, when they are exposed, they expose a lot of natural flora and fauna, they tend to get accumulated, and that accumulation can eventually impact the microbiota of these flora and fauna, which eventually can come down to us, and that these problems are actually real. So not only the pathogenic bacteria which we think about can become resistant to a particular antibiotic that we get worried about because we are no more going to get treatable with that. But environmental bacteria, as we just saw, can also become resistant to these antibiotics. So should we worry about that, or should we not worry about that because they are environmental bacteria and we are not concerned about them because they do not cause us any kind of disease? One required to remember here that ecology is very closely knit. It has a flow, and any kind of change that occurs in the ecosystem For example, let us think that antibiotic can have some direct effect on bacteria. It can reduce its population size. It can have a very costly resistance in the form of evolution. That would mean that it really cannot propagate very well. It can have a very elevated high GT levels, is the horizontal gene transfer levels. As you people, many of you must be knowing that bacteria can exchange genetic materials amongst within a community, and that is called horizontal gene transfer. There can be an increased rate of mutation because of the antibiotic pressure. There can be alteration in the cell signaling. We will talk about biofilms later, and also a change in quorum sensing. That would mean that it is the way bacteria communicate within the environment changes. Now, all these, even if it is from let us say an environment, non-pathogenic bacteria, what potential effect can it have on a species interaction? And that is something which we should not ignore, because it can change the population dynamics due to reduced population sizes. And I'll give you an example of it. Effect on bacterial community composition. It can have an altered trophic interaction. It can also change eventually the ecology of the area. Now, to just give you an example, you may have read in your textbooks that we on our skin has a number of other bacteria living, which are actually not infectious, which are not pathogenic, but they are our natural flora. Now, if many a time they discourage the colonization of pathogenic bacteria by sheer competition, and just think for a moment that this is the ecosystem we are talking about, and this ecosystem gets an abuse of antibiotic, killing a number of such bacteria which are otherwise fairly harmless. There are two kinds of threats that we can anticipate. It gives the pathogenic bacteria a chance to colonize now. Alternately, if pathogenic and non-pathogenic bacteria are co-colonizing, and the non-pathogenic bacteria has an antibiotic-resistant gene, there is a possibility of its horizontal gene transfer to the pathogenic bacteria. So something which is actually happening in nature possibly is not going to remain restricted to nature, and we should open up to that. So far, what we were trying to say is that antibiotics, which were discovered for treatment of various kind of ailments, they targeted very specific physiological events of bacteria, and therefore were able to make them either dead or could limit their growth. They could be broad spectrum, they could be narrow spectrum, but because of their abuse, we eventually find antibiotic resistant emergence of antibiotic resistant bacteria. So bacteria which were earlier treatable are no more treatable, and I'm talking about pathogenic bacteria to mankind. And of course, there are a lot of antimicrobial resistance to the bacteria which causes livestock diseases as well. So, and we also saw that these antibiotics, when they are in the environment, they find their way, even if they lead to, due to their pressure, can lead to resistance in non-pathogenic environmental bacteria, which can again get transferred to pathogenic bacteria. And there is a possibility of changing in the ecological system, which definitely is a disturbance, and we should wake up to it. Now so we have been talking about development of antibiotic resistance. Let us quickly see that how the antibiotic resistance actually develops. It's a very natural process. It's a very natural process that if you have, it's a natural process of adaptation. It's a natural process of natural selection. So this is mainly for the students who are listening. So if suppose you have an antibiotic treatment, and amongst the population of antibiotic, there are two which are natural resistant strain. If you use the antibiotic, possibly these two strains will live and the rest will die. And eventually, even in the presence of that antibiotic, the antibiotic resistant strain will proliferate. this is more because more happens when you use suboptimal concentration of antibiotic. Remember, the impact of antibiotic is also very concentration dependent. So if therefore every dosage has a particular concentration, every dosage has a duration. So if you take them suboptimally, then it gives a chance of already existing in the population certain natural mutants which are resistant, a chance for them to proliferate, and then they become overwhelmingly on that particular population. It can also happen that the non-resistant bacteria in the time of proliferation actually gets a mutation which becomes beneficial for it in adapting to the antibiotic pressure, as I said, and then the drug-resistant bacteria can multiply. And here I will take a pause and I'll ask the students, if you want to experimentally do it in your labs, you can actually do it in your lab and see that whatever we talk about, all these things that we talked about, that suboptimal use of antibiotic, improper use of antibiotic, expired antibiotics, rampant use of antibiotics without discrimination, whether all these things can actually lead to antibiotic resistance or not, can be very nicely shown in your own labs. You can actually do an experiment where if you, and I think I suggest that if teachers can make them do this, and they will see that if you grow a uniform based population and then keep growing them in suboptimal concentration of antibiotic, and eventually you will find that some of the bacteria are independent colonies that are growing, and then you can go for its genetic and phenotypic characterization, and you will realize where the mutation or what kind of property this particular bacteria has, which is different from its parent population, that would tell that yes, this is the phenomena that occurs in nature as well. And you will be surprised to know the number of generations that they require to acquire this, which is not which is not very long. Similarly, you can also have other ways of finding out that if in particular, say, there are antibiotics which are particularly targeting an enzyme, and you know the target enzyme, so you can do the inoculation and you can keep passaging them, and eventually when you find a molecule, find a bacterial isolate which is now resistant to the antibiotic that you started with, you can specifically study only that particular gene, only that particular enzyme, and study what all mutation has accumulated in this particular the target of the antibiotic, because of which the antibiotic is no more functional. So what I'm saying is, whatever we said that is happening in nature is not that it does not have any experimental evidence. You can do this and experimentally show that yes, suboptimal usage are improper uses, or antibiotics which are expired can actually have an impact on development of antibiotic resistance, and the bacteria which was otherwise getting killed no more gets killed. So not only that, we are talking about one particular bacteria, one particular population, but the problem here is the transfer of the antibiotic resistance. Okay, so that put some of the other points here. So when people were trying to study, the transfer of antibiotic resistance, they mostly what they do is they take the clinical samples and then they try to see that they do the DNA sequencing, and they try to study the genes underlying this, and then they do epidemiological dynamics. Okay, so if a particular isolate is resistant to a particular antibiotic, people try to see that if they can trace that resistant the genotype of the resistant strain, and see that from where it has really where it has come. But antibiotic resistance can be transferred, and the mechanism of, and you can go to any textbook, because I will just give you certain points and you can actually look into it. Of course, one way of antibiotic resistance is the spontaneous mutation that can accumulate in a specific target gene or target protein, which makes the antibiotic no more active, no more working. But at the same time, the antibiotic resistance gene or antibiotic resistance factors of from a strain can be also transferred to this another strain in the form of mobile elements. Now, you all must have these mobile elements are generally can be plasmids and can be also through bacteriophages. So you can have conjugation based plasmid transfer, and this particular plasmid has high has a gene which causes, or what you can say is that it attacks the antibiotic and disables it. So therefore, it is a resistant gene, and that can be transferred through conjugation from a resistant strain to a non-resistant strain. It can be also passed through transduction by bacteriophages. It can be also through transduction, where the plasmids can be taken up by this, but they are generally transferred when a resistant strain is communicating or is in a, rather is in a community where there are a lot of bacteria resides. Okay. Here, one other thing that is very curious, and I think I'm telling mainly to this to the student, is that once this is an area of intense research, and you people should think about that there we still don't understand the basic mechanisms of how antibiotic resistance are transferred in a community, and one can think about many options, especially how are they evolutionary conserved. One must understand that we understand when the plasmids or whatever is acquired is preserved if there is an antibiotic present. The generation of antibiotic resistance, actually the bacteria requires to pay a price in the terms of either there's a growth limitations or there's a virulence limitation, but it has also been seen that they overcome these. And eventually there are compensatory mechanism, evolutionary mechanism, compensatory evolutionary mechanism at the molecular and cellular level that occurs, which permits the bacteria to maintain an antibiotic resistance gene despite no antibiotic in their environment. And I think it is important to understand a lot of basic biology that can be done in understanding the transfer of antibiotic resistance, and that is a very exciting area of research. So the compensatory mechanisms, if we understand what are those which permits the bacteria to retain such kind of thing, possibly can help us think about a very different way of tackling antibiotic resistance and permitting it to not be transferred from a resistant to a non-resistant bacteria. Yet another factor, so yet another factor which contributes to antibiotic resistance is biofilm formation, and this is a very, very very stubborn factor which contributes to it. And here it's generally biofilms, they are comprises of syntrophic consortium of microorganisms. So there are syntrophic microorganisms, there are different kind of microorganisms, different phenotypes of microorganisms that can exist here, and they release a kind of polysaccharide or polymeric substance which makes matrix around it, which prevents its penetration. The moment antibiotic penetration gets prevented, you know that the optimal concentration of antibiotic is no more there. The antibiotic, suppose you require 100 mg, 100 microgram of per ml of antibiotic to kill a bacteria. By the time it penetrates to the lowest level of this, it will remain maybe 10 or maybe so. It's so suboptimal that it allows the persistence of those which can persist, and although they do not replicate. Then you have a certain kind of bacteria which can release certain enzymes which will neutralize the antibodies, which will degrade the antibodies. Some are slow grower. So biofilms are very stubborn. Biofilms are generally a consortium of microorganisms which are syntrophic in nature, and is a major problem in case of hospitals where you have stents and you have catheters where there is a biofilm of bacteria generates. So because of the biofilm, because antibiotic cannot be optimally reached to kill, and that can also lead to generation of antibiotic resistance. But this is a natural process, and we require to figure out how we can penetrate these biofilms. So there are other ways by molecular mechanisms by which bacteria develops this resistance. And you will find some very beautiful reviews on this, where biochemical mechanism, as I talked about, bacteria can mutate its target, and therefore the antibiotic no more targets the, is not able to work anymore. It can have enzymatic inactivation of the drugs, like for example, beta-lactamases are an example of that, where it can degrade the beta-lactam antibiotics. Sometimes they can acquire genes which are less susceptible to be targeted from other species. Sometimes they can bypass target. For example, I can give you a vancomycin. So vancomycin generally goes and attaches to de-alanine peptide, pentapeptide chain when cell wall synthesis takes place. And what the resistant bacteria does, it changes it to de-alanine and D-lactate. Now, vancomycin can no more attack it. So they try to make sure that they bypass the target, and then they can become resistance. Some of them preventing drug access to target is equally interesting, because there are efflux pumps that can throw out the antibiotic that is worked in. So now if you think about this whole scenario, you can think about the number of areas that are open for research, because we do not have the entire picture of bacterial physiology, which can be very specific to a specific kind of bacteria. It can also be very generalized for many bacteria. Now, the idea is to identify those specific and those generic mechanisms so that we can design an antibiotic accordingly. So far, what we have done quickly is that we have seen a brief on antibiotics. We saw how antibiotics work, and because of suboptimal use, how the emergence of antibiotic resistance has occurred and how we have arrived there. And eventually, we know that if you do not use sufficient concentration of antibiotic, how the bacteria naturally gains its resistance by developing various mechanisms at molecular and cellular levels, and even at community level. Biofilm is a community level of developing resistance to antibiotics. Now the last point of how can we fight it, or rather how are we fighting it? And at this point of time, we should not think that this is the end, or should we think that this is the end of the modern medicine, because many of the antibiotics are, many of the bacteria are already resistant to first-line antibiotics. And that would mean the treatment requires to be longer, treatments will be toxic, it will increase healthcare and is huge economic burdens on families and societies. At the same time, if you think about such kind of resistant bacteria everywhere, we are going to be worried about organ transplants, chemotherapy, and surgeries. And once again, in just 75 years, within the span of 75 years, because of our indiscriminate use, we have hastened, we have process of antibiotic resistance, and we hope we are not leading to this pre-antibiotic era, which was very, very scary. So what are we doing about it? Now I'm going to give you certain examples of what are the various ways that we are trying to tackle. Okay, and these are the area which are area of very active research. There is a void. We can do better. We are not doing enough possibly, because we still require a lot of search and research to be done. And that too in the area of finding new antibiotics. Alternative to antibiotics, should we think about it? And definitely in case of communicable diseases, and what we have seen in the case of COVID, prevention and control possibly is a huge area where, so if people are listening who are not from science background, there's a lot that infectious diseases can there are a lot of research that can be done, because there's a huge socioeconomic impact of infectious diseases that always occurs. Rather, in the examples or in some of the discussions that will come after this, you will realize that no single person, no single area. It has to be a very integrated approach to address the issue of antimicrobial resistance if you have to find out new antibiotics and alternative to antibiotics. Okay. To fill up the void, let us see what all can be done. And one of the area which is very, very intensely being worked out is to discover the untested new chemical diversity. We believe that nature has a lot more chemical entities that we know. There are research labs which are just concentrating on cataloging those structures. They do it by NMR, they do it by, they fractionate it by HPLC, and they do a lot of NMR structures, and they are just trying to find out what is the chemical entity which even possibly we are not able to synthesize and we do not even know about it. So amongst them, a continuous effort to discover natural product diversity there are very interesting research that is going on both in India and abroad, and marine and aqua sources, as well as sources like endophytic bacteria which are endemic to a particular region, are being explored for their for possible antibiotic. For example, recently there is a yellow marine pseudomonad which was found to release a new quinolinol compound, and in Germany they also found a new member of pluramycin class of antibiotic. Pluramycin class of antibiotic are not only antibiotics, but they are also used as anti-cancers because they inhibit nucleic acid biosynthesis, and they found it from a marine Streptomyces species. So there is a lot of scope in actually exploring the natural products and to find out new antibiotics. Not to say that there is also a lot of scope that is there in synthetic drugs. One good example of synthetic drug is the very recently introduced TB drug for XDRs, the extreme drug-resistant tuberculosis, that is called linezolid. It is a synthetic antibiotic which is, it disrupts bacterial growth by again stopping the protein synthesis, but it does it by a different mechanism, and so therefore cross-resistance to other antibiotic is prevented. But here I would like to tell you, and maybe you should go back and also check the papers there, where very recently in MIT, a machine learning platform has been used to discover new antimicrobial compounds. This particular system, which also uses artificial intelligence, has screened more than 107 million chemical structures, and they have found there are 9 potential antibiotics. Amongst that, there is one antibiotic which actually could take care of 97% of the drug-resistant bacteria, and that's very promising. Not only that, the same, I think the model was then repeated with the Broad University at in a project with MIT, and they again screened a library of 6,000 odd structures, where many of these structures were in preclinical stages and all, to find out they have come with a specific particular molecule which they call it as halicin, and that is again a very promising antibiotic, which gives us a lot of hope that there is still a scope of finding very good molecules in order to tackle antibiotic resistance here. The target-based approach is yet another area where many of you would like to contribute. Target-based approach is like first understanding the bacterial system itself. You can have, so there are a lot of omics-based studies that you people are aware of. The people do study the changes in the profile of transcription and protein levels, and finally at the metabolite levels, which we call as metabolomics, to understand how a bacteria behaves when it comes under infection stress. Infection, when it does, it is not a very conducive environment for bacteria. It's a very hostile environment for bacteria, and bacteria really adapts, rewires its metabolic network in order to survive. So that area itself is an area of intense research, understanding the rewiring of transcription, translation, and metabolism by bacteria in order to find out the choke points, in order to find out molecules, factors which are essential for survival of bacteria, is a very, very area of active research. People do both phenotypic as well as genotypic screens in order to find out a specific. So phenotypic screens do not give you a target, but when you do understand the system and find out a specific enzyme, then you can use that enzyme, you can study its structure and actually find out if you can design an analog that can be specifically targeting it. A recent example, although again from TB I can give, bedaquiline is a good example, which targets the ATP synthase of Mycobacterium tuberculosis, and which is a very critical for generation of ATP and eventually the survival of bacteria, and has been approved for MDR, for XDR treatments. Yet a very recent one has targeted polyketide synthase. Polyketide synthase 13 is the terminal polyketide synthase, which is important for mycolic acid synthesis in tuberculosis, and for that they have identified a heterocyclic molecule which is called thiophene. Okay, so there is this target-based approach is a very good, very vast area, very interesting area to work and find out new targets and validate new molecules. An interesting thing, another interesting way of identifying new antibiotics is repurposing of old drugs. Now here, I believe that if I can shift for a little bit from bacteria to even viral at the present day, what we are trying to take care of in COVID-19 is repurposing of drug. You must have heard that hydroxychloroquine is being attempted to be used for the treatment. So repurposing of drug means a drug which was earlier being used for some other purpose and some other system is repurposed for it being used as an antibacterial. So, or antibiotic. Here I'm giving you the example of the one which has been approved for COVID-19, that is remdesivir and also favipiravir, which both of them are RNA, they inhibit RNA-dependent RNA polymerase and had been tested in other viruses and have been approved by US and Japan, and possibly we are also going to try to see if especially favipiravir, whether it can be repurposed and it can work in our population as well. But coming back to the bacterial example is one of the example that is clofazimine. Clofazimine is being used for leprosy before, and it's being tried to be repurposed for MDR XDR TB, and people are attempting to see if it will work against them or not. So repurposing of drug is beneficial because most of these drugs have FDA approval, and therefore they have been already proved to be less toxic or not toxic for human consumption. So that saves a lot of time in the process of drug discovery. Yet another way of finding a new molecule is rationally improving the existing antibiotic, and this is very interesting, and students who are listening possibly can do this exercise. So how do you rationally improve an existing antibiotic? So for example, you know the structure of an antibiotic. Let us take an example. Let's say that, okay, I take rifampicin. We know the structure of rifampicin or any other antibiotic, and we also know the target of that particular antibiotic. We know that RPO beta is the target of it. Now we also know that the resistance comes because of accumulation of mutation in this particular gene, or which changes the structure of the protein itself. So one can now take help of a lot of computational tool to see that how a mutation can change the structure of the protein in such a way, not impacting its function, but impacting the interaction of the antibiotic to it. And then once again can try to fit the antibiotic and try to find out what will be a rational way of adding or removing a side chain that can improve the existing antibiotic. So of course it has to go through the process of, once again, any change has to go through the entire process of toxicity checking and all, but that's another way of thinking, and that anyone can, person who has a fairly good understanding about computational tool can attempt to see if these things can be done or not. And the last are, that's what we are going to discuss, but not the least, of course, it's a very interesting thought that people are thinking about selecting a new antibiotic. Is saying, can we go back to the old antibiotic which has been taken off the shelf? See, the reason I was telling you that antibiotic resistance has come, that eventually it has been not properly used, or naturally the bacteria has developed a resistance to it. So now suppose for 40 years the antibiotic has not been used in the market. Is it a possibility that the bacteria which were resistant to this antibiotic no more exist, and therefore the bacterial population, which is the pathogenic bacterial population, is actually sensitive to such old antibiotics? So people are giving it a very serious thought and say that if there is, we can rediscover whether the old antibiotics, which has not been used for 40-50 years, whether they would work in the present scenario. If not alone, can they be used as an adjunct? And one of such antibiotics is octaptin, which was discovered 40 years ago and has been forgotten by scientists altogether. And it is being thought of being used for some of the superbugs these days. And maybe you would like to read a little bit more on it. So besides discovering of new antibiotics by various ways, so we have multi-pronged way of trying to find out new antibiotics. We also have to find out alternatives to antibiotics. So yet another few area where very interesting work is going on. Of course, the work is going on finding vaccines, and we already have quite a few vaccines which are protective vaccines. We are still searching for a vaccine for several of the viral diseases, and COVID is one of them. And people might be more aware of what is happening in case of attempt to search for a vaccine, because several attempts are being made to use attenuated strains of coronavirus 92, as well as even India has a particular strain which was isolated in Pune and is going to be they are trying to see that if that can be a vaccine candidate. While protective vaccines are fairly, you might be knowing about them, there are very few therapeutic vaccines. So there is another wing to vaccines which is called therapeutic vaccines, which can be actually used along with antibiotic, especially against those which are antibiotic resistant varieties. So therapeutic vaccines, we do not have any approved therapeutic vaccine other than, I think, FDA approved CVAC for cancers. But the therapeutic vaccine, which relies on the fact that it can boost the immune system in such a way that can help further the antibiotics to clear the pathogen. So this is another area which is fairly at its infancy, and any new thoughts, any new kind of idea is going to be really useful in making therapeutic vaccines. Another area is antimicrobial peptides. If there are postgrad students here, and also antimicrobial peptides are natural cationic peptides, and they function by virtue of just interacting with the very negative cell wall of bacteria, and they penetrate, making holes into it, and therefore disrupts their osmolarity and causes the death. They are part of innate mechanism, and therefore they are not very selective, as broad spectrum as it can be. So, and people are now searching for antimicrobial peptides as an adjunct with the various kind of therapy, antibiotics. So antimicrobial peptides can be yet another answer to an alternate to the solution to antimicrobial resistance. Although knowing bacteria, knowing natural evolutionary sequence, we should not be surprised that they will eventually become resistant to these as well. But our search, we have to find a process in such a way that we are ready with a modified molecule, even anticipating that there will be a resistance to this particular molecule. An area which is at its infancy but is really picking up is bacteriophage therapy. Had it been in any other platform, I would have actually asked the students, so how much have you heard about this bacteriophage therapy? bacteriophage therapy, as you all know about, phages are the viruses which attack bacteria. And the use of such phages to treat bacteria is not very new. It is actually a century old almost. However, there are no currently FDA approved bacteriophage therapy for treating human pathogens, but this is definitely there for food industry, where they use it bacteriophages to biocontrol of bacterial pathogens like salmonella, like staphylococcus, listeria, pseudomonas. So in food industry, they still, because they are biocontrols, they are used as, but there are very interesting. So bacteriophage are very specific in nature. They are very, very specific. So one bacteriophage will go and attack and kill a bacteria, a very specific bacteria. So what concerns are about bacteriophage therapy possibly is that we do not understand the nature of immune response that it will elicit. But some of the recent trials, in 2016, there was a trial on drug-resistant staphylococcus, multi-drug-resistant staphylococcus aureus infection of human diabetic foot ulcer. So there was topical application of the specific bacteriophage against it to 6 patients, and all the 6 patients recovered, and that's very promising. That similarly, there was some phase 1 or phase 2 trials of antibiotic-resistant Pseudomonas aeruginosa infection of ear infection, where also there are very promising results on application of bacteriophage as against the placebo. So there is a huge potential of bacteriophage therapy in finding as an alternate mechanism of dealing with antimicrobial resistance. So these are some of the other ways by which antimicrobial resistance can be tackled. Each of them are a research area. Each of them are very challenging. Each of them have a translational value. Each of them has lot of basic science to understand. Each of them have very integrated approach. You cannot be just a biologist or just a chemist or just a physicist or just a computational biologist to do this. You have to come up as a team in order to understand and in order to address such a question. Not to mention that there are also immunotherapy which are equally important and is an equally good and possibly a sustainable area of research. The value of immune health has been very well defined now, especially in case of what we see as a COVID pandemic, and also the fact that many of the bacteria, even if I talk about tuberculosis, if we say that out of 100 people who get infected, only 10% develop the disease, that means that our immune system, those 90 people, they either they never develop the disease despite of infection, is only because their immune system is sufficiently strong to contain the infection and to develop the symptoms. Same is the case of the present pandemic that we see. A large number of people are recovering by virtue of their immune strength, and therefore strengthening your immune system in various forms like nutraceuticals. Nutraceuticals are those food which can be taken, which has additional value than the normal nutritional value. Of course, you have a huge area of work which is going on in probiotics, where people are trying to understand. They also can generate engineered probiotic microorganism, which will by various mechanisms can get rid of antibiotic resistant or pathogenic bacteria, or can by sheer competition or by maybe releasing secondary metabolites there do not permit the infectious agents to harbor in you. So these are another set of area where people are thinking about finding new ways of tackling antimicrobial resistance. So very quickly, so this is what are some of the ways that we can fight antimicrobial resistance, and we are trying to fight antimicrobial resistance. Very good leads, but much to do, both in the terms of basic as well as applied research. So my urge to all the students who are listening, if you are thinking, and you should be thinking about a research career, you should join these searches and researches. Each of them, depending upon your interest, pick up, read more, and you will be able to find, or you will be able to design very nice projects around these problems, both basic and which can have an eventual translational value. So now, to just possibly, I would like to end with a message that I've been trying to give to many, that at an individual level, what is that we can do to prevent this antimicrobial resistance to develop as fast. Remember, antimicrobial resistance is a natural phenomenon. It is almost like if I am put into a stressed environment, I will try to adapt and survive. So that is a natural thing that happens with all organisms. So similarly, if there is an antibiotic in the environment, the bacteria will try to resist, and there will be certain population which will be able to resist the antibiotic, and that population will be selected by natural selection, and there will be resistance. So all said and done, antibacterial resistance is a natural phenomenon. But at the same breath, we can say we can delay the process, and that is what we have not done. So if you require to delay the process, there are certain things that you always require to remember. At our own personal level, don't misuse antibiotic. Finish the course, follow the dosage, prevent infection. The prevention is always better. We always say that prevention is better, and that is what is being advocated even in the present pandemic, where they say that hand hygiene, keeping yourself clean, making a distance so that you are not in close proximity with the disease or the infected person. All these are maintaining health and maintaining hygiene. The kind of food you take to the students, the stress factor is a huge factor, and that is important that you get properly and maintain your immune health so that you can prevent getting an infection. And if you get an infection, make sure you take the right kind of medication. If not required, especially in kind of things like flu and all, you really do not require a medication, and you can tide over it in maybe 1 week or maybe 10 days, and eventually your immune system should be able to take care of it. When you take an antibiotic under certain conditions, you end up destroying your own natural flora, which are helpful for you, and that actually can give way to other infections, which I think we should be really, be careful about. And I urge that you spread the word. Although must add that the present crisis of antibiotics is not only for abuse and development of drug resistance, is also that we really require to have very stringent policy. For example, we require to have very strict policies about use of antibiotics in livestock. We have to have involvement of pharma companies, because initially the way pharma companies were interested, they do not really come forward, which have become very, very short. Means very soon, because of our own indiscriminate use possibly, or the antibiotic resistance strains are coming at a frequency which keeps a new molecule, the moment comes in a few years, the shelf life is gone, its life is gone, because there is a resistance to it already. So that discouragement would also require and actually help us in overcoming those crises. So I'm not going to discuss about those, although they are a new area of debate and discussion. Today, I will just keep my discussion only to the scientific research part one in tackling the antimicrobial resistance, how we arrived at it, and how we can prevent it, and also how we can contribute in finding new molecules to tackle this problem. So remember, the antibiotic resistance problem cannot be solved, it can only be managed. With this, I'll stop my discussion and would be happy to take the questions. Thank you.
Thank you, ma'am. Is it audible?
Yes, it is.
Yeah. Thank you, ma'am, for enlightening us with your wonderful lecture and emphasizing on emergence of drug resistance and explaining experimental approaches for drug resistance, and also addressing antibiotic bioaccumulation problem. So having known that, still we are trying to solve the problem of antibiotic resistance. We have to manage with alternative to antibiotics like natural plant products and probiotics. Ma'am, really your lecture is enriching us, and you have shared your knowledge with us. We are really thankful to you on behalf of Bhavan Vivekananda College. Now the session is open for question and answers. Dr. Y. Aparna will be putting the selected questions questions from chat session. We will give opportunity for one or two speakers to directly talk to Dr. Sharmistha. Thank you, ma'am. Over to Dr. Y. Aparna.
Thank you, ma'am. There are many chats telling for thanking you for an informative lecture, and they were almost everybody has posted that it is a very nice, elaborative, and informative lecture. Thank you once again, ma'am. There are n number of questions posed, but I'll just ask you some 3 to 4 questions. So,
Sure.
one is by Gayatri Ramachandran. She asked, is this broad spectrum or narrow spectrum that is majorly responsible for MDR globally?
See, a comparative study, possibly to what I understand, has not been pursued to find out which one is more responsible for it. Okay, but what happens if you use a broad spectrum antibiotic instead of where a narrow spectrum can work, that it ends up killing many friendly bacteria in your body. So when friendly bacteria kill, as I was telling you, that gives an opportunity of the pathogenic bacteria to harbor. And once such kind of pathogenic bacteria harbor, they again intermingle with a source which can have an antibiotic resistant element, which can be horizontally transferred. Okay. A narrow spectrum antibiotic case can be used if you know that for sure that the source of infection is only this. Otherwise, it many a times may not work. And because it may not work, that is the fear why intermediate spectrum antibiotics are given generally. So comparing which one is going to contribute more, using narrow spectrum antibiotic is less of a concern, but a broad spectrum antibiotic very rampantly without any kind of diagnosis being used can definitely cause harm to very friendly bacteria.
Thank you, ma'am. We have one more question from Ushasri. Madam, if we change the formulation of existing antibiotics, can we reduce the rate of antibiotic resistance?
The rate of antibiotic resistance will depend upon a number of things. So if it is a particular targeted, it's a antibiotic, it targets a specific factor, and okay, so how much, with what rate the mutation accumulate in that target protein. So when you are finding a new target, that is one of the criteria people think about, that if I'm finding a new target in bacteria, I'm going to look for certain features of that. One is low mutation rate. Another is that it should not be present in the host, so that the molecule that I designed for it doesn't go and cause harm to the host. So, and the low mutation rate is important because that would permit it not to very frequently change. So there are certain very basic enzymes which do not mutate very frequently, and that's why if you have seen the way antibiotics have been working so far are against the basic physiology. Now, what the question you asked is that whether changing the formulation will change the rate of resistance has to be worked out. yes, maybe certain specific formulation like using multiple combination of drugs can make sure that none of the resistant strain, within the population stay. If you are changing the structure of the molecule by addition of a new functional group or new side chain, that can also become a new molecule. But whether that particular functional group is targeting an area which is less mutable in the bacteria is an important thing that you should think about. Yes, the answer is yes, it can be reduced, but it requires to be worked out whether the kind of formulation you are changing will change the rate of development of resistance or not, because development of resistance depends upon a lot of factors. If you titrate the dose well, maybe you can also arrive at a dose which will delay the development of resistance. Yes, that can be done, but that requires to be really worked out.
Yeah, we have one more question from Dr. Mohan. Can we make an obsolete antibiotic a repurposing and used against AMRs?
Sorry, come again. Obsolete?
Antibiotic.
Obsolete.
Obsolete antibiotic repurposing and used against AMRs.
Yes, I think that is a possibility, because that is one of the thoughts that people are having, to rediscover the old molecules. The molecules which has not been used for several years, 40, 50 years or something, and the molecules which have been not in the environment, in the distribution for some time. People are again trying to use those molecules and try to see either if they are existing antibiotic this work or not, or if they can be repurposed for the purpose of antibiotic resistance. I gave you an example of octapectin. You search on that and people are actually thinking of using octapectin, which is some 40 years old molecule. People almost had forgotten, and they think that it can be recalled to the front line of to tackle superbugs. Yes, that's a possibility. In fact, when we are trying to search for antibiotic, we should just try to explore all sources possible.
One last question, ma'am, here. Dr. K. Vindhya Roy has posted, what antibiotic policies are in place in India to control antibiotic usage?
You are asking the wrong person. I will not be able to tell you about the kind of policies that are there, but I am sure ICMR has a very strict guideline for usage of antibiotics. That's the reason I did not talk about policies, because it's a discussion in itself. ICMR has very strict guidelines for usage of antibiotic. See, implementation becomes a problem possibly, and that's the reason when I only talked about what we can do from our side, because rest of the things are really in our hand. Yes, government should be pressed for implementation, for proper policies. There are very strict guidelines on what all can be sold over the counter. There are very strict guidelines whether broad spectrum can be used when, which special. So if you pick up any for these policy documents, let it be TB or something, there are very clear guidelines. It's just that implementation somewhere it suffers, and because of which it's not that we don't have clarity on what we require to do. I think that the soft point is the implementation, and the only thing that what we can do from our side is adhere to certain things which are within our range. I'm sorry, I'll not be able to tell you more about policies, but more than policy, I feel implementation is the one which is suffering.
Okay, thank you, ma'am. One very interesting question which we are facing very recently. People are asking, like regular usage of sanitizers and 70% alcohol, can they bring resistance? Because we are using it regularly on our hands. So can they bring resistance?
I think it's a good to think about it, because I had something and then in my slide, and then I thought I'll remove it because it will create a confusion, because the mandate these days is hand hygiene. So so regular, I would say that use your there are reports. So let me tell you that there's a report in one of the Canadian journal, which says that overuse of disinfectant at home has led to differences in the microbiota of children, and some children have shown tendency of obesity. Okay, so you will find all kind of reports everywhere. So I'm sure things are connected, not that those reports are incorrect. There are correlations like that. So what I would suggest at this point is, the idea of maintaining hand hygiene is, especially when you are trying to touch with those hands your mouth or nose, or where the infection can get inside. And so try to make sure that you clean your hands when you require to do that. Not for every small thing, that suppose I opened my door and got in the home itself and I feel that it is contaminated and I should start rubbing my hands. No, don't do that. Overusage, you will find that it is going to be bad for even the normal cells on the surface of our skin cell. So it has been told to maintain hand hygiene. It has not been advised to overuse it. Now there's a lot of things that there's a very thin line between that. Just think, pause, that suppose you have taken your garbage bin outside and then you come back in. It is important that you wash hand. And that is not only for COVID. I mean, who told us that we have to wash hand for only today? We have been told this since do not know when by our parents, that when you come back home, you wash your hands and feet, and that hygiene has been maintained. So I suggest that yes, we require to do it, all the more because now there is this uncertainty about this particular virus. But these kind of measures will take care of many diseases, be it diarrheal disease, be it any other kind of viral disease. You will find that these kind of hygiene measures are applicable for prevention of most of the diseases. And overuse of sanitization, don't overuse sanitizer. If you already know that there is an overuse of sanitizers at 30%, of course, after some time there are already reports, not in context with what is happening today, but people have already reported about overuse of disinfectants. We tell that don't overuse bleach into your commode or something, because when it goes for decomposition, there are bacteria there which can die. So these are very normal things that we are aware of. So my suggestion to you would be that don't overuse. Use logically when you are about to use your hands or something, for when you're taking them close to your face, or you think that you are using a very general article which can be used by several people. So if it is being used by several people, for example, when I use my own laptop which nobody touches, I don't keep sanitizing my hand. Okay, but if I use a very general place, something, after usage, I possibly will sanitize my hand. So I think these are something which you have to think and make sure that you balance the two, so that there is not an overt usage either. For the time being, it will not cause antimicrobial resistance, but it definitely is going to harm the skin on the surface. So that is what I do.
Into the cells.
Yeah, I mean, they are not used to 70% alcohol. They are not evolved to do that. So,
yeah. Thank you, ma'am. We have, dear participants, whatever the questions you people have asked, we can't ask everything right now, but definitely we will get the answers from the resource person. We will pose the questions to madam, and once we get back this, we will come back to you.
So I have something to tell students, especially those who are interested in a research career. If you have any questions if you're thinking about research career in infectious diseases and you have any questions related to that, feel free to contact me by mail. They will have my mail address. If I can answer some of your questions, or the areas which are very active area, very challenging area, very interesting. I see all the areas are equally interesting. And if any one of you have a question related to your research or with the kind of research going on, or feel free to ask me. Feel free to send a mail, and I'll try my best to contact you and give you the answer.
Now, any one faculty and one student can directly interact, because time.
ऐसा नहीं है कि जहाँ पे लोग आए
Sorry.
आपका earphones.
Please, sincere request. Please, one by one. Only two. A sincere request. Please raise your hand. It's better. Please raise your hand. I'll unmute them. That is much better. Momin Shakir. Momin Shakir, you can ask the question. Kajal, you can ask the question.